preparing your sample to be put on the sequencer, and then all the way on the other end with tools for data analysis. So we partner with small companies that have advanced solutions that either improve the workflow, the data accuracy, or they help save money. So here you might recognize companies such as Covaris, Archer, Lexogen, and then today we're going to be focusing on Lexogen for RNA-Seq. Lexogen is a biotech company founded in 2007 and headquartered in the lovely Vienna, Austria. So this company is solely focused on supporting the RNA-Seq workflow with the high-quality library preparation, and then recently also with data analysis. In 2014, they expanded to the US, and they partner with DMARC, and we represent them in Canada. So we're going to talk about their unique proprietary technology that enables detailed analysis of either the full transcriptome or gene expression studies. So today's objective, to provide you with an update on the recent advancements in RNA-Seq technologies. So our first focus is going to be for whole transcriptome sequencing, so how to achieve better data with a more advanced library prep kit. And this is using the Sense technology. Our second focus, for those of you who are just interested in gene expression analysis, will be on 3' UTR-seq. So this is a very cost-effective way to count genes using RNA-Seq. So it removes that cost barrier that could be preventing you from moving from microarrays to RNA-Seq. So let's start with the whole transcriptome sequencing part. So what are today's issues in library prep technology? The first and foremost is quality. So the workflow hasn't changed much in years. Many kits still involve that fragmentation step, which is crazy. It's magnesium digestion, and you're adding heat to your RNA. This is very damaging to the RNA. Followed by a reverse transcription, which can affect your strand specificity. And then, of course, PCR, which can introduce bias. Second is time. As we all know, time is money, and these protocols are quite long and can take a few days to complete. Lastly, price always being an issue. Usually these can be between $50 and $80 per prep, and that's not including the poly-A selection or your ribosomal depletion. So I'm going to talk about how Lexogen's solution can address all three of these issues. So here's a visual of your standard RNA sequencing workflow. So as you can see, they all involve the fragmentation step. Here's 3 different ways. You have Illumina RNA ligation, your DUTP method, and then your RT method. So they all have your fragmentation, your reverse transcription, and then your it's easy to visualize how fragmentation and PCR can affect your data quality, but not many people are thinking about the reverse transcription step. So I just wanted to go into detail of how RT artifacts can affect your data. The main issue occurs during first-strand synthesis, where spurious second-strand cDNA can be created. So this can happen in 2 ways. The first, as the strand is elongated, it can flip, self-prime, and then create this hairpin structure. I just want to note that fragmentation can only add to this issue as it adds more 5' ends that are available, so it just makes it more likely for this structure to occur. Secondly, the strands can displace, prime, and then create second-strand cDNA. So this spurious second-strand cDNA can be interpreted as a true antisense transcript when in fact it's not. So why is strand specificity so important? It is known that more than 1/4 of human genes overlap, so that's quite a big number. 10% participate in bidirectional promoters. So below here is a link to that reference in case you want to know more information. But if you're using a kit that does not preserve the strand orientation, then you run the risk of detecting false genes. So here we have an example of 2 overlapping genes. If you use a proper strand-specific kit, then the genes will be called accurately in data analysis. But if you don't, then here are just a few of the possible combinations that could be incorrectly called during data analysis. So this slide shows the difference in data that you get from the overlap enough and RP1 genes. So the top will show accumulated coverage that you get if you use a non-strand specific kit. So the bottom two will show two different coverages on both the minus and the plus strand that you get if you use a strand-specific kit. It's important to note here that the coverage on the minus strand is 2 orders of magnitude lower than on the plus. So here you see the numbers from 1 to 11 versus 1 to 27 on the scale. This essentially means that transcription is much lower for the minus strand. So this highlights the importance of having a sensitive, high level of strand specificity in your library prep. So now that I've outlined all of the current issues in library prep, how does Lexogen help? Well, here's their secret sauce, and it lies in their Sense kit. They have a starter-stopper heterodimer mix, which is shown here. So you start with your poly-A mRNA bound to your magnetic beads. The heterodimers will then bind randomly along the transcript. Reverse transcription will create your first strand of cDNA from the start to the stop site, as shown in this red box here. So when it reaches the stopper, it's efficiently stopped, and the cDNA fragment is then ligated to the stopper. So essentially, you can completely skip the fragmentation step, as the insert size will be dictated between the start and the stop site. So then you go into your second strand synthesis, and then finally your PCR amplification, and here you introduce your barcodes and then your adapters for cluster generation. And this is all in a single tube protocol. So as a result, you get greater than 99.9% strand specificity. So how does this differ from kits that claim 98%? Well, it turns out, as I mentioned before, the antisense transcripts are much less abundant than sense. So really that 2% difference is quite significant. So here we have some RNA sequencing data showing sense and antisense reads for each annotated gene. With 98% strand specificity, as you can see, you're only getting 40% of the antisense transcripts. Whereas with Lexogen sense technology, with 99.9%, you're getting, in this case, 100% of the transcripts detected. But we typically see this number to consistently be at least 99.9% or greater. Because of this streamlined protocol, the workflow is also much shorter. So here we compare Lexogen's Sense kit to Illumina's TruSeq protocol. So Illumina's protocol can take a few days to complete, whereas Lexogen's kit can be done in 5 hours with under 2 hours of hands-on time. So I'm sure those of you who have done the TruSeq protocol know the pain of how long it takes. I mean, even reading the protocol can take forever. So the Lexogen workflow is much shorter. So this is data based on the sequencing of samples from the US FDA Seq-C project, or the sequencing quality control. Oops, get rid of that. This is the accumulative coverage using the Sense kit in green, and then in this case, we compare to TruSeq in blue. So as you can see, Sense has more even coverage across the transcript, whereas TruSeq will show a 3' bias. So just to recap on the value proposition that the Sense kits can offer, and it covers quality, time, as well as price. So firstly, the poly-A selection kit is included at no extra cost. If you're doing total RNA-Seq, then they also have a ribosomal depletion kit available. Their patented starter-stopper heterodimer mix allows you to skip the fragmentation step. The reaction is also quite efficient, which means that you can get away with less PCR, which of course reduces the PCR bias. Importantly, the kit has excellent strand specificity. The workflow is quite fast, 3.5 hours if you're doing total RNA-Seq, and then 5 hours if you include that poly-A selection. Lastly, the kit is cost-effective at $36.50 Canadian per prep for 96 samples. So let's move on to the second topic of the day for those of you doing gene expression analysis. If cost is an issue that prevents you from switching from microarray technologies to RNA-Seq, then you'll be really interested in this new technology. It's called QuantSeq, and it's also offered from Lexogen. So we like to break RNA-Seq into two major types. You have your whole transcriptome sequencing. So here we create multiple fragments per transcript. So this is great for applications like de novo assembly, isoform and fusion detection, and gene expression analysis. And then you have your expression profile sequencing. So here you only create one fragment per transcript. So this is only useful for gene expression analysis. So as you can see, whole transcriptome seq requires 10 times the amount of reads. So here, on average, it will take 100 million reads versus expression profile sequencing, where now we're getting into only 10 million reads required. So if you're just doing gene expression analysis, whole transcriptome can be a bit of an overkill. First of all, it's a waste of sequencing reads. And secondly, in data analysis, it consumes a lot of computational resources. So the advantage that you have with expression profile sequencing is that since you can get away with less reads, you can multiplex more samples on the sequencer. And that's where the real cost savings lie, as you can multiplex up to 96 samples in one lane. In addition, the data analysis is much less intensive. So you can get away with faster aligners, and there's no need for any RPKM or FPKM concentration calculation. And here's a nice overview of the technology. So you start with your mRNA transcript. An oligo-DT primer will then bind and extend to create your first strand of cDNA. During your second strand synthesis, the primers will bind randomly along the template. So the one that binds closest to the 3' UTR end will successfully extend into that first primer. And then this will create your double-stranded cDNA library, which will then undergo your PCR amplification, where your barcodes and your adapters are introduced for cluster generation. So this way, only one fragment per transcript is sequenced, and that fragment will be adjacent to the 3' UTR end of the transcript. There are two versions of the QuantSeq kit. So the first is what I just mentioned, the QuantSeq 3' mRNA-Seq library prep kit. They also have the QuantSeq Flex for a more targeted approach. So I'm just going to focus on QuantSeq 3' first. And here's a comparison of the QuantSeq expression profiling data with standard mRNA-Seq. So this graph will plot the relative coverage across a normalized transcript length. So the mRNA-Seq data is shown in gray, and then you have your QuantSeq data shown in green. And then here's your ERCC control in the dark gray over here. So as expected, the QuantSeq coverage will be focused more on the 3' end. But what's important to note here is that you're getting the same number of genes with both methods. Additionally, in this case, 12 times less reads was used for QuantSeq than for standard mRNA-Seq. So this graph essentially validates QuantSeq as a viable alternative to standard mRNA-Seq for your gene expression analysis with much less reads, which allows you to multiplex more. So as I mentioned before, you can multiplex up to 96 samples per. This dramatically lowers your cost per sample. So in this case, we use the cost of library prep. So here we have $28 per sample from a 2x96 kit. So let's say you want to multiplex 48 samples. So it's 1,344. The cost to run one lane of a high-seq 2500 in Canada on average is about $1,500. So in total, that'll be $2,844. So this works out to $60 per sample for expression profiling. And if you really want to take advantage and multiplex 96 samples, then this can be as low as $44 per sample. So from what I'm told by customers, it's not unusual if they're using standard RNA-Seq for these costs to be well over $100 per sample. So you can really see where the cost savings lie. So let's turn our attention to the second version of QuantSeq, and this is QuantSeq Flex for a more targeted approach. So if you have any specific sequences along the transcript that you're interested in, then this is the kit for you. You can combine the QuantSeq 3' and the QuantSeq Flex kits. So this gives you the ability to tailor your experiment according to your needs. So QuantSeq Flex gives you the opportunity to use either target-specific primers instead of oligo-DT for your reverse transcription and random primers during your cDNA synthesis. So you can do your reverse transcription with oligo-DT, followed by either a random or targeted second-strand synthesis, or you can use targeted reverse transcription followed by either random or targeted second-strand synthesis. So this is useful for those of you doing applications such as fusion detection for novel fusion. So to sum it all up, QuantSeq is the best sequencing-based alternative to microarrays. So the library prep is very cost-effective at $30 a sample. In combination with the sequencing cost that you save through multiplexing up to 96 samples in one lane. It's an extremely reproducible kit and quite sensitive for low abundant transcripts. Short protocol with 2 hours of hands-on time, and you can input as little as 100 picograms of RNA. It's a very robust kit, works very well with low-quality RNA such as FFPE samples. And lastly, the data analysis is simple and can be performed without any sort of bioinformatics experience. In fact, Lexogen has recently teamed up with Bluebee to include the data analysis pipeline in the platform at no extra charge for any QuantSeq customer. So what I was mentioning before, if you can do $44 per sample, that includes for 96 samples of multiplexing, that includes not only your library prep, includes a sequencer cost, and it also includes data analysis since it's at no charge. So when you buy a QuantSeq 3' kit, then you get an access code, and then this is used on the Bluebee site for analysis that's at no charge to you. So the data analysis can be done by any user, and you don't need any bioinformatics experience to perform it. The data is processed in 10 minutes, so it's very fast. If you'd like to know more information, feel free to contact myself, and I can put you in touch with Lexogen to further discuss. So how does this work? You simply register on the Bluebee platform using the code that's provided with the QuantSeq kit. Then you connect your sequencer to the platform, you upload your data, you run your analysis, and then once processed, you retrieve your results. It's very simple and easy to use. So the QuantSeq data analysis is grouped into 3 major steps. So here we have your quality control steps, your steps for alignment, and then lastly your steps for gene counting. And all these steps are incorporated onto the Bluebee platform. So if you're a QuantSeq user, you get a free, fast, and very efficient data analysis tool at no cost. No bioinformatics experience required in order to do the QuantSeq analysis. So the standard Bluebee setup includes reads trimming with BBDUK, alignment with STAR, and then counting with HT-Seq. So if you're interested in third-party publications of researchers that are using QuantSeq, Lexogen's website is a great resource. So just go to lexogen.com/publications. Here I just have a few examples of the recent studies that have been shown in 2016 using QuantSeq. Just go to the website for more info. Here are just a few examples of publications using the Sense technology. Okay, so before I open it up to questions, I have a few announcements. Lexogen is currently having their year-end promo, so they're giving 10% off all products as well as some gifts, and this is valid until the end of the year. So feel free to contact me if you would like a quote. If you want to try any of these kits, we do have 4 prep samples available at no charge. So again, contact me for more info. Lastly, Lexogen has just released their dual indexing capability, and this is available for both Sense and QuantSeq. So now you can multiplex up to 384 samples using this technology. Okay, so thank you for tuning into our webinar today. So at this time, I'd like to know if you have any questions. We do have our Lexogen technical specialists here who are happy to answer any technical questions, and that's Katerina. And then we have myself, again, my name is Erica, and I'm here to answer any logistical questions. So thank you very much.
Thank you very much, Erica, and thank you for introduction. So here is me, Katerina, and Yumlin, who is our senior application scientist, and we are happy to answer any of the questions which you may have about our product. If you have a question, you can type it in the chat window, and we will just answer them. Okay, we have a question from Ram. The question is if we can perform splice junction analysis. Could you please a little bit elaborate? Are you talking now about Bluebeam solution or data analysis for SENS? Maybe. I see that you don't have a microphone involved, so maybe you can type a bit more about this question.
So just in general, maybe. So with our SENS kit, the whole transcriptome analysis kit, you can also detect splice junctions. Yes. So this would then be probably the SENS kit that gives you the whole transcriptome information. Once the kit is really dedicated to generate fragments of the 3' end, so if you expect to have something in the 3' end, close to the 3' end, you will still get it, but it's not for the whole transcriptome. And then there is another question from KK. If there's still both versions of QuantiQ, forward and reverse, and yes, so still both versions are available. The standard kit we recommend is the forward kit. Yeah, and the reverse kit is what we recommend if you're really interested in a 3' UTR. So it starts directly at the 3' end of the transcript, and therefore you need a custom sequencing primer to kind of cover the sequence of the tail, because otherwise you would need to read through the whole tail, and then you would have a problem with cluster calling in the sequencing. But this is also included in the kit, so it comes with the kit, and you just need to replace the standard Illumina multiplexing primer with the custom sequencing primer, and it should work as normal.
Reverse kit is also recommended in case you are interested in paired and sequencing. Do we have any other questions? Okay, then if we don't have more questions, we would like to thank everyone for joining this webinar. And thank you, Erica, for this good introduction of the product. And if you have any questions about SENS or QuantiQ, please feel free to send your email or contact Erica. And we are happy to help you also with the test kit if you're interested.
Thank you, Katerina and Ermelind. And as well, I just want to also thank everyone for joining the webinar today. Have a great day, everyone.
Thank you.